let us look into the demo of the Izhikevich neuronal model. So first, let us cover a brief background about the neuronal models that we are seeing right now. We have seen during the last classes. So the brain networks work in coordination in order to generate the behavior. And computational neuroscience tries to mathematically explain how the behavior is generated. So once the mathematical model is ready, we can implement it using the computational tools. So computational neuroscience provides the tools and methods for characterizing. So broadly, based on that, we can divide the models into what models, how models, and why models. The what models are descriptive models. They will address what the nervous system do, and how models will are mechanistics models, and they tell us how do they function. And the why models are interpretative models, so they will answer the question why they operate in a particular way. So now, so the understanding the brain using the computational models. So these descriptive, mechanistic, and the interpretive models. So we will be looking into the mechanistics model here. So how we can simulate the behavior of a single neuron on the computer, and how do we simulate the network of neurons. So that will be our focus for today's session. So we can see that this is a very complex task. And the 300 cross 300 millimeter square of a mammalian thalamocortical surface, it consists of 100 billions of neurons and 10 to the power 5 synapses. So this is a functional description of the mammalian thalamocortical surface. So how do we model that? To go about it, we will see how the modeling, these different levels of modeling can be done. So there has to be a trade-off between the detail and the speed, and the computational traceability and the abstraction. So we are ultimately looking at a very large networks of neurons, but we cannot skip the important single neuron dynamics and the picture of a whole network, which can show the behavior. So let us partition the model of the individual neurons depending on the details it can show. So in that way, we can model it into level 1 models, that is the detailed compartmental models, which we saw using the cable theory, that it describes the complete morphology and the spatial structure. So they are very realistic, and they will be able to model all the dendritic numerical integration. Then they can model the axonal behavior. They can do the discretized dendritic numerical integration. But they are the disadvantage is that they are computationally very complex. It is extensive, and it consumes a lot of time. So if you are looking at modeling a whole brain or a behavior aspects, so this level this detailed level of modeling will be consuming too much of time, and we may lose a lot of behavioral details in between. So let us look at the another level of the modeling, that is a level 2 reduced compartmental model. Here we are reducing some of the complexity, but it is sufficient enough to showcase some of the somatodendritic interactions. It will retain the multiple time scales and the dendritic interactions. And but it is still it is used to model the short-term memory and the oscillations, the slow wave sleep oscillations, etc. But it is still computationally complex, whereas the level 1 model was used to model a single neuron. This can model up to a network of 300 neurons. So next level is the level 3, that is the single compartmental model. Here the main focus will be on the ionic currents and the production of the voltage, that is at the subthreshold and the spiking voltage traces. So we have seen a broad class of networks describe this phenomenon, like Hodgkin-Huxley, the integrated fire neurons, and the Izhikevich neurons. So they model the characteristics of the spikes, that is phasic spikes, the tonic spikes, and the different other varieties like adaptive spiking and the other cell characteristics that are observed during the spiky behavior. So this is computationally less complex, and let us look into a few more levels of abstraction. So level 4 is the cascade model. So if we are looking at the interworking between different areas, so cascade models will be helpful, particularly in the visual system where the simultaneous adaptation to the mean light intensity and the contrast and other features can be combined. So it involves concatenation of linear filters, linear, non-linear transformation, and the random processes. So this is a little more abstract version, and this is a rate-coded model. And next one is that the final level of abstraction is at the black box level. That is, it doesn't care about the details, intrinsic details that is happening inside the box. What it will deal with is that given an output and provide what is the input provided, it will come up with the function that is underlying in between. So but it the disadvantage is that it loses too many information in between. So it is not good for modeling some kind where we need to look into some specific subsystems where details are significant. So having the background of all these modeling levels, let us try to find a, now important thing is to find a balance, whether we have to go for a detailed approach or whether we have to go for an abstract approach. So where we draw a line in between. So as a complex system should be like a good caricature. It should emphasize the important features and it should ignore the non-essential features. So so now the brain, like we know that the brain function relies on the interplay of millions of neurons, billions of neurons. So, and each neurons will have will be displaying different sort of dynamics and behaviors. So how do we selectively take care of some properties of the neurons and ignore the other properties of the network? So that can be done only by understanding how single cells operate as part of the network. So every cell will be behaving differently when connecting to the network. So there are different kinds of mechanism that is exhibited by a single cell, and that is what we will be looking at today. So we can briefly summarize 20 ways to spike the neuron. So that is where the neuronal models like Izhikevich, which you saw in the last class, we can display wide varieties of spiking behavior, all by controlling 4 variables. So the first one we are showing is the tonic spiking. That is a regular spiking in response to a step input in current. So it is a periodic in nature. It is periodic spikes in response to the step input. Now, next one is the phasic spiking in response to the step input. Then the tonic bursting. So we can see that it is a periodic bursting. It bursts for a small amount of time and then it continues in a regular periods. Then the phasic bursting. It bursts for some time, then it is silent. And now the mixed mode. So sometimes it bursts, sometimes it doesn't burst. It is periodic in nature. And the spike frequency adaptation. So like how does the spike frequency changes over the time? And then next is class 1 and class 2, where in the class 1 it fires at a very low threshold, very low current threshold, and the spiking the frequency will be proportional to the current. In the class 2, it requires even higher current, but the thing is that it fires at a constant moderate frequency. So, and there is another variety where it is a spike latency. There is a delay which is inversely proportional to the current, and the spike timing, it is all encodes the strength of the input. Higher the input, the spike timing will be dependent on that one. And there are sub-threshold oscillations. So the neuron does not only give an action potential type of response, it also exhibit the sub-threshold oscillations. So we can control our parameters in order to exhibit the sub-threshold oscillations. Then the resonator. It is the spikes happens when the current input resonates with the subthreshold oscillation. When the peak of the subthreshold oscillation matches with the input provided, it will resonate and then a spike is provided. Then there are integrator. which without subthreshold oscillation, we can integrate the inputs that is coming from multiple dendrites and uh, depending and with a specific frequency. Then the rebound spike, this is about the inhibitory inputs. So now we have different kinds of inputs can be excitatory or inhibitory. So on reception of an inhibitory input, the uh, spike is generated from the when the inhibitory input is released. That kind of behavior can be modeled. And the rebound burst, so now earlier is a spiky thing, now this is a burst thing actually. So a burst of input spikes are produced in the from the release of the inhibitory inputs. Now again another variabilities, that is threshold variability. Inhibition lowers the threshold and excitation raises the threshold. So there is a variability of threshold for the spiking. And the bistable nature, which you have already seen in some of the neuronal models. And then this is depolarization after the potential. That is, it may experience a period of super excitability after a spike instead of a refractory period. And then there are other varieties called accommodation. A slow increase in current gives neuron time to accommodate. And inhibition introduced spiking, and the inhibition induced bursting. So that, as name indicates, we can see that how the spiking and the bursting data behavior is seen. So now, how do we model all these behaviors of the neuron? So which neuronal model will be helping us to model that one? So most of the neuronal model can exhibit some of the response patterns, not all. And some of these patterns are mutually exclusive. That is, they are similar. And they are different actually. So now, Izhikevich neuronal model help us to model all these 20 types of behaviors by that is by controlling some of the configuration parameters. So other neuron models come close. They exhibit some subsets of the behavior. So let us look into the ways to model the neuron. So this is, you are already aware. So V is the membrane potential. Then we need how the voltage potential is varying with respect to the time. And the input I is the input current. So voltage typically is in millivolt range, and the time is in millisecond stage, millisecond scale. And the integration step, we use the Euler method. So now this is a tabular for data that summarizes all sorts of models and what they are good at representing. So there are a wide variety of these dynamics that can be illustrated, that is biophysically meaningful, that tonic spiking, phasic spiking, tonic bursting, etc. So, and the left column will indicate show the integrate and fire, and the up to the Hodgkin-Huxley, which is a detailed biophysical model. So Hodgkin-Huxley is able to describe all sorts of behavior because it is a very detailed biophysical model, and it is computationally expensive also. And you can see the Fujii-Nakuma model, which you are currently doing in the assignment, and the Izhikevich model, it can display almost all types of patterns. Now, this is the flow chart. This is a illustration like that there is a trade-off between the biological plausibility and the computational efficiency. So the biophysical model, detailed biophysical model, it is expensive in time and least computationally efficient. And the next model, the Izhikevich model, which is close to describing all the biophysical model, but it is a little bit reduced in terms of complexity. And then the leaky integrate fire and oscillatory model, which you already seen, and the rate coded model that is at the highest level of abstraction. And now this is okay, I have to hide. The implementation cost versus the biological plausibility, and the various models are mentioned there. So now this is how we model the Izhikevich model. So it is a dynamical system. The important variables are V dot and U dot, and the V dot is given by this equation, that is a quadratic equation, and this U dot is the recovery variable. U is the recovery variable, and that is given by that equation. Now you can see that in this, if the voltage, okay, let me take out the pointer. So the voltage, initial voltage, when if V is equal to 30 millivolt, so when the voltage reaches its peak, we have to reset the condition. The voltage is brought back to the reset condition, that is at C. So when it is brought back to the reset condition, same time what we will do is that the U variable, that is the recovery variable, that will be uh, uh, changing from U plus D is tending to U. So that decays at a slower rate, and the rate of decay is A, and then finally it is attaining the level which is having a difference of D. That is the reset difference. So this is what we will be modeling and describing all these 20 types of behaviors. And this is the typical mammalian thalamocortical surface. The red dots indicate the excitatory spike, and the black dot indicate the inhibitory spike. So this is a raster plot. So we can see that the spiking nature. And so this is a network of 512 cross 512 neurons. So dynamics. So it is very hard to understand the dynamics looking at the cortical level recordings. So these are the references used for this presentation. And so finally, I have just highlighted few of the parameters so that we can implement that in our model. So let us go to the demo part. So this is the website. You can find the details about the spiking model of the model of the spiking neuron. If you want details, you have the PDF file, you have the tutorial program, and the even the MATLAB code is given. So we'll be using that to simulate some of the bursty behavior, the spiking behavior. So let us go into that one. So this is the code. This is a very simple code. It uses only 4 parameters actually. So and then directly that equation. It looks longer because we have different parameters. Each parameters for different spiking, that is a tonic spiking, phasic spiking, and so on. So if we run that one, so this is the output of the program. So now we are comparing, we are modifying this dynamics using the 4 variable, that is A, B, C, D. So we will see few of the spiking behaviors. Let us go into the phasic bursting first. So that is indicated, A variable is given by 0.002, and let us configure this 0.25 minus 65. And then you can see that the current is provided, and then you can see the phase plot also. Let me run it. Let me close it and then run it again. and then let's provide the current input. So we can see that it is a phasic burst, phasic spike. So, okay, let me maximize the window. So the second one is a phasic spike, the one shown in B. I'm not sure whether that is visible, so that you can see in this. Because this is the phasic spiking. So now let us modify that the parameters in order to exhibit a tonic spiking. So only thing what we need to do is that we have to change the variable here. You all can download this program from that website, and let us give a current here. So now you can see that the behavior is changed. So this represents the tonic spiking. And similarly, let us look at a phasic burst. So for that we need to modify the parameter. Let me remove it. Yeah, so this is the phasic burst. So it bursted, and then it was silent for some time. So and the next burst, and giving a current. So the next burst will be coming. So I don't know how to increase the speed of. Let me see the visualization speed. Let me increase so that it will move faster. So here, this bar you can control the input current, and here you can see the modify the visualization speed. And then here you can these buttons with these buttons you can control the excitatory and the inhibitory input, and also you can modify the initial points in the portrait. So now this is about the phasic burst. Let us see a couple of few more, two or three more spiking behaviors, and then we will look into the Morris-Lecker model. So let us go with a tonic burst. The parameters are 0.02, 0.25, yeah, so now it should exhibit tonic bursting. Yeah, so that is so now there is another thing. Now we saw the tonic response and the phasic response. Now there is another aspect, that is a mixed mode. Sometimes it is tonic and sometimes it is bursty in nature. So in order to model that one, we have to change the parameters slightly. Yeah, so this is tonic burst, and then now so yeah, so this is the mixed burst. Right. So now let us also look into that spike frequency adaptation in order to. So the current is given, it is spiking when the current is withdrawn. So there is a lack of spike, and then again, so there is an adaptation between the nature of the current given and the frequency of the spikes. So there are a few more that we have covered around 7. Like there are 20 different behaviors which can be simulated with. I've summarized everything. Like only thing that you need to do is that in order to generate all these things, all the 20 different types of behaviors, we need to. So these are the different parameters that we need to provide to the model in order to generate it. That is A, B, C, D, and then I. That is the input current. So this is for the next set of 10 bursts. So I think remaining you can explore on your own. Like, is there any doubt related to this one? Then we can address that. Otherwise, we will look into the Morris-Lecker model.
It's fine.
Okay, fine. So the program, you can, if you are interested in exploring it further, you can. Maybe I will paste that link in the chat so that if someone has missed out on that one, then they can look into it. I will. So this is the MATLAB code, and everything is attached in present in that file itself. So now let me show the demo of the Morris-Lecker model. So before that, let us go into some theoretical background so that we'll have a recap. So I will directly show it in that notes, that NPTEL notes. So you all have been modeling the Fujin-Akumo model. So in that one, what you can see is that there is the difference in this is the Fujin-Akumo model null clines. So this is one is a S-shaped null cline, and another one is a straight line. And then intersection of these null clines generate is contributing to different kinds of fixed points, and which can be stable or unstable. So this is a bistable. This Fujin-Akumo model exhibits bistable behavior. So now, what is the difference between Fujin-Akumo model and the Morris-Lecker? So this is, I think, this is all this is already covered in the class. Morris-Lecker model. So it consists of 3 kinds of channel. Let us take out some of the key points of this model. So it consists of a fast activating calcium current, and it is having a delayed rectifying potassium current, and then it also consists of a leakage current. So now this is a simplified model. This, as in case of FN model, the dynamics of M is fast, and that can be replaced by M infinity, and the equations of the ML are given by the what is shown below. So again, so this is the calcium dynamics, this is the GK1 is the potential dynamics, and then the GL into V minus EL is the leakage dynamics, and the M infinity and W infinity are the gating variables. You can see that finally the dynamics is described by the membrane rate of change of the change of membrane voltage with respect to time. So that is given by the equation above. That is c dv by dt plus the equation phi to 1. That will describe the dynamics of the system. And now this is the stability of the bifurcation plot of the Morris-Lecker model. So initially it will be stable up to here it is mentioned 0 to 68 picoampere. I tried to code this particular model and I observed the stability at around 88 picoamperes. So I don't know whether there were some parameters I ignored, but I am seeing it at 88 picoamperes. And from 88 picoamperes to 220 picoamperes, here it is 220 to 217, mine it is 220. We can see limit cycle oscillation, and after 220 onwards, we will see the stable behavior again. So these are the null clines, the types of null cline. One will be a inverted N shape. So V null cline is an inverted null N shape null cline, and W null cline is a sigmoid. And now the behavior of the response of the system depends upon the intersection point. On which point of the V null cline does the sigmoid, the W null cline intersects? So if it is intersecting on the first, let us divide this V null cline into, okay, let me take that up. You can see my cursor or not able to see the cursor? I think, okay, only I can see that cursor. So let me.
Oh, we can also see so.
Okay, you can see this cursor. Okay. So in this, if the intersection happened in the first segment, so it will be stable behavior. And if it happened in the second segment, then it will be a limit cycle oscillation, and here it will be an again the stable behavior. So this is the voltage response. So now let us directly go into the model, that is the implementation. Yeah, this is the limit cycle behavior. We can see oscillations, and then when it is going to the third region, you can see again a stable behavior. So let me run the code. So okay, so first let me describe. Yeah, this is the variation of the current. You can see that's the applied current. So as the current is going from we saw from 68 up to 0 to 68 picoamperes, it is a stable region. But here we can see up to 88, it is stable. And then now we enter into a limit cycle. You can see, so this is the starting point of the trajectory. So the trajectory goes on, and then it will end up into a limit cycle. So we can see the oscillations that is happening. The top right one is the membrane potential. The middle one is the gating variables. Green is W, and other one is the M gate. So now it crossed that stable limit cycle period, and then it is reaching the third segment of the V null line, and it is again going into the stable mode. So this is the dynamics of the Morris-Lecker system. So this is actually, this consists of these are the variables that, okay, you can also see that you can also observe how the when this dynamics are happening, how is the, so it mainly it consists of the calcium and the potassium dynamics. So how the potassium and calcium currents are varying with respect to the current that is applied. So main variables are V1, V2, V3, V4, and then the calcium conductance, the potassium conductance, and the leakage conductance, the applied current. And so if you want to show that in a loop, you have to run it in a loop, and then use the differential equation solving using the Euler method, and then you can compute that. So it is just a two-line differential, 2-3 line differential equations. So you can let me done. So this is initially, it is starting at minus 10, 0. That is the starting point of V is equal to minus 10, and then W is equal to 0. That is the starting point. It is coming, and then it is reaching that fixed point. That is the intersection of the two null lines. And suddenly when it crosses that threshold, current threshold, it enters into a limit cycle until some point around when that until the applied current is 220, and then afterwards you can see. And now this is the calcium and the potassium currents, how that is varied across time. And this is the gating variable variation across time. For convenience, I've shown only till 0 to 200 milliseconds. Yeah, and it reaches the stable region. So this is the Morris Lecker, and then similarly you can try, if you have time and then if you're interested, you can try out the other simplified models, which will help you to understand and learn better. So this is the this is another variation of the simplified neuronal model, INAPK. And yes, so that is it from the demo session. If you have any doubts, then we can address that. Otherwise, we can end the